You know that feeling when you're staring at an exam question and you're just like, what do they actually want from me here? Well, today we're going to pull back the curtain on all that. We're taking one single question from a real biology paper and we're going to break it down piece by piece to show you exactly what an examiner is looking for to give you those top marks.
So, here's our game plan. First, we'll dig into the experimental setup. Gotta know what's going on. Then, we'll look at the results and figure out what they're telling us. After that, it's time for the deep dive into the hormonal explanation. We're talking auxin. And to wrap it all up, we'll crack open the mark scheme to see how you can get every single point. Sound good? Let's jump in.
All right, first things first, the setup. You know, in science, how you design your experiment is just as crucial as the results you get. It's all about asking the right questions in the right way.
Okay, so take a look at this. This is the whole investigation right here. We've got 3 different setups. In A, the light is only coming from one side. Then you have B, which is completely in the dark. And C gets light from all directions. Super simple, right? But these 3 conditions are the key to unlocking this entire question. Everything hinges on understanding what each one is for.
So right off the bat, we get a classic question. They're asking for control variables. Basically, what things do you need to keep exactly the same for all 3 of these plants to make sure your test is actually fair? Go on, have a quick think. What are a couple of things you'd list?
And here's what the mark scheme says. You could have mentioned the same volume of water, the same temperature, using the same type of plant, or even the same age of the shoot. Any 2 of those would get you the marks. And it makes perfect sense, right? If the temperature was different, or one plant got more water, that could totally mess with the growth rate and ruin the whole experiment.
But wait, here's a really important little detail from the examiner. The mark scheme says yes to water, but it specifically says do not accept light. So why is that? Well, think about it. Light is the one thing we're changing on purpose to see what happens. That makes it the independent variable, not a control variable. Getting that difference is absolutely key.
Okay, let's talk more about these controls. It's a really common mistake to just think a control is, well, the normal one. You don't do anything to, but it's way more specific than that. Each control here, B and C, has a very distinct purpose. So the question is, what are those purposes?
And here is the answer, word for word, from the mark scheme. Let's look at B first. By keeping it in the dark, you're proving that the growth response we see in A is actually because of light. Simple. Now look at C. With light all around, the plant grows straight up. This proves that the bending is caused specifically by light coming from just one side. It's a subtle but crucial difference. That's the level of precision they're after.
All right, we've nailed the setup. Now let's shift gears and actually look at the results. What do the plants themselves tell us? What can we figure out just by observing?
Okay, zero in on experiment A for a second. See those little ink marks on the shoot? At the start of the experiment, they were all spaced out evenly, but now, not so much. The question wants to know what 2 things you can conclude just from looking at how those marks have moved.
The clue is all in the spacing of those marks. Look at the area right behind the very tip of the shoot. See how the marks are really spread out there? That's your first point. Growth or elongation is happening right there. And hey, where is the shoot actually bending? In that exact same spot. So there's your second point. The bending happens just behind the tip too. It's literally just describing what you see. 2 observations, 2 marks. Nice and easy.
All right, this next one is a quick vocabulary test. You've done the hard part. You've described what's happening and where it's happening. But what's the proper scientific name for this response? What do we call it when a plant grows towards a light source? And the magic word is phototropism. That's it, that's the mark. And it's easy to remember if you just break it down. Photo means light, like in photograph, and tropism is just a fancy word for a plant's growth response to a stimulus. So, phototropism is a growth response to light. Simple as that.
Here's another one of those little examiner insights that can make all the difference. You could also say positive phototropism and get the mark because the shoot is growing towards the light. But, and this is important, if you wrote negative phototropism, that would be wrong. So that little detail about the direction, positive or negative, really matters.
So we've covered what's happening, where it's happening, and what it's called. Now it's time to zoom in and look at the how. We're moving from the big picture, what we can see with our eyes, to the microscopic level. We need to talk about the hormone that's pulling all the strings, auxin. To get to the bottom of this,
The experiment introduces a new element. We have two more setups. F is just a normal chute tip. But look at G. They've inserted a tiny little glass barrier right down the middle of the tip. Both of them get lit from the side, just like in experiment A. So the big question is, what difference does that little piece of glass make?
Okay, let's walk through the 3 points you need to make here. First, in the normal chute, F, the auxin hormone, collects on the shady side, the side furthest away from the light. Got it? Okay, point 2. In chute G, that glass barrier physically blocks the auxin from moving across, so you end up with an equal amount on both sides. And the final killer point is putting it all together. This proves that auxin doesn't just sit there, it actively moves sideways away from the light, right there in the tip of the chute.
Now for the final part of the question, and this one tackles a huge, super common misconception about auxin. People often wonder, does the light actually destroy the auxin on the sunny side, or does it just cause it to move over to the shady side? Well, this experiment answers that. What can we figure out by comparing the total amount of auxin in all the samples?
And here it is, the big reveal. The mark scheme points out that all the samples have pretty much the same total mass of auxin. This is so important. It means light does not destroy auxin. All it does is make it move. It migrates from the bright side to the shaded side. That higher concentration of auxin on the shady side makes those cells grow longer and faster, and that is what causes the whole chute to bend towards the light. Myth busted.
Okay, let's pull back and look at the big picture again. We've learned a ton about how plants bend towards light. But maybe more importantly, we've learned a lot about how to think like an examiner.
So here are the 4 big takeaways. Number 1, know your controls inside and out. Don't just know that they're controls, know why. Number 2, use the right words. Phototropism gets you the mark, just bending won't. Precision is everything. Number 3, stick to the evidence. The question about the ink marks was about the ink marks, nothing else. And number 4, explain the how. It's not enough to say the plant bends. You need to explain the whole auxin story. It moves to the shade, which causes faster growth on that side, which leads to the bend.
See, when you really break down just one question, you learn so much more than just the facts. You learn a whole way of thinking. It's about being precise, backing up your claims, and connecting the dots. So I'll leave you with this final thought. Now that you've had a peek inside an examiner's mind, what's the first thing you're going to change about how you revise?